Have you ever wondered how scientists can study thousands of proteins from a single biological sample? Every cell contains an enormous number of proteins, and these proteins are constantly being produced, modified, degraded, and interacting with one another. The field that allows us to study this complex protein landscape on a large scale is called proteomics. In this video, we will take a step-by-step journey through proteomics, starting with how genes produce proteins and then moving through protein extraction, digestion, liquid chromatography, mass spectrometry, and finally protein identification using computational analysis. By the end, you will have a clear understanding of how modern proteomics experiment works. To understand proteomics, we first need to understand where proteins come from. The basic flow of genetic information is represented by the central dogma of molecular biology. DNA is first transcribed to produce RNA. Then RNA is translated to produce proteins. So, the overall flow is DNA to RNA to protein. But here is the important part. Proteins are the molecules that actually perform many of the functions inside a cell. They participate in metabolism, signaling, transport, structure, defense, and many other cellular processes. And the relationship isn't as simple as one gene producing one protein. Through processes such as alternative splicing, along with different modifications after translation, a single gene can contribute to multiple protein forms. This enormous diversity is one of the reasons why studying the proteome is so important. So why can't we simply study DNA or RNA and understand everything happening inside a cell? The answer is that having a gene doesn't necessarily tell us how much of its corresponding protein is actually present or what that protein is doing. Proteins are the functional molecules driving processes such as metabolism, signaling, and immunity. Importantly, the amount of messenger RNA doesn't always directly correspond to the abundance of the protein. A protein may also be modified after it is produced, activated or deactivated, transported to another location, degraded, or involved in interactions with other proteins. Therefore, if we want to understand the actual functional state of a biological system, we need to look directly at its proteins, and that's where proteomics becomes extremely powerful. So what exactly is proteomics? Proteomics is the large-scale study of proteins present in a cell, tissue, organism, or a biological sample. It doesn't simply ask which proteins are present. Modern proteomics can also ask how much of each protein is present, how do protein levels change between conditions, what modifications have occurred, and how do proteins interact with one another. The proteome is incredibly complex. Even though the human genome contains roughly 20,000 protein coding genes, the number of distinct protein forms can be much larger because of processes such as alternative splicing and post-translational modifications. And unlike the genome, the proteome is highly dynamic. It can change in response to nutrition, disease, stress, environmental conditions, development, and many other factors. So proteomics gives us a dynamic picture of biology at the protein level. Now let's look at the overall workflow of a typical proteomics experiment. Everything starts with a biological sample. This could be cells, tissue, blood, food, microorganisms, or another biological material. The first major step is protein extraction. Once the proteins have been extracted, they are prepared for analysis and typically digested into smaller peptide fragments. These peptides are then separated using liquid chromatography. After separation, they enter the mass spectrometer, where they are ionized and analyzed according to their mass to charge ratios. The instrument also fragments selected peptide ions to generate MS-MS spectra. Finally, these experimental spectra are compared with theoretical spectra derived from protein sequence databases. This allows researchers to identify peptides and ultimately determine which proteins were present in the original sample. So the overall journey is sample preparation, protein extraction, digestion, LC separation, MS-MS analysis, database searching, and protein identification. Let's now go through the workflow step by step. The first stage is sample preparation. If we are working with cells, the cells need to be disrupted to release their intracellular contents. This process is called cell lysis and can be achieved using mechanical or chemical approaches. Once the cells are disrupted, we need to extract the proteins while removing unwanted components such as DNA, lipids, and cellular debris. The extracted protein mixture then needs to be quantified. The common methods for measuring total protein concentration include the Bradford assay and the BCAA assay. Why is quantification important? Because we want to start the downstream analysis with the known and reproducible amount of protein. Good sample preparation is critical because problems at this stage can affect every step that follows. After extracting and quantifying the proteins, we reach the next important step, protein digestion. Mass spectrometers are generally not used to analyze intact proteins in a typical bottom-up proteomics workflow. Instead, proteins are enzymatically broken down into smaller fractions called peptides. One of the most commonly used enzymes is trypsin. Trypsin preferentially cleaves proteins after the amino acids lysine, represented by K, and arginine, represented by R. This produces a complex mixture containing thousands of peptides. Each peptide represents a small portion of a protein. These peptide fragments are much more suitable for liquid chromatography and mass spectrometry. So we can think of digestion as converting a very complex mixture of large proteins into a collection of smaller, more manageable molecular species. These peptides then become the molecules that we actually analyze using LC-MS. Now we have thousands of peptides, but there's a problem. If we put all of them in the mass spectrometer at exactly the same time, the instrument would have difficulty distinguishing all of these signals. That's why we use liquid chromatography or LC. In many proteomics workflows, reversed phase liquid chromatography is used. The peptides interact with the chromatographic material according to the properties such as hydrophobicity. A solvent gradient is then used to gradually elute the peptides from the column. As a result, different peptides leave the column at different times. This produces a chromatogram containing distinct peaks. The major advantage is that the complexity of the sample is reduced at any given moment. Instead of thousands of peptides entering the mass spectrometer simultaneously, smaller groups of peptides enter over time. This improves detection and helps mass spectrometer achieve deeper coverage of the proteome. So LC acts as an important separation step before mass spectrometric analysis. Now we arrive at one of the most important technologies in modern proteomics, mass spectrometry. Before peptides can be analyzed, they need to be converted into charged ions. This can be achieved using a technique called electrospray ionization or ESI. The charged peptide ions then enter the mass spectrometer. In the first stage, called MS1, peptide ions are measured according to their mass to charge ratio or M by Z. Selected precursor ions can then be fragmented. This produces smaller fragment ions. These fragments are analyzed in the second stage called MS2 or tandem mass spectrometry. The resulting MSMS spectrum contains characteristic pattern of fragment masses, and this pattern provides information about the sequence of the original peptide. So, in simple terms, MS1 tells us about the precursor peptide, while MS2 gives us fragmentation information that helps us identify that peptide. After the mass spectrometer finishes collecting data, we have a huge amount of information. But the instrument doesn't simply display the name of every protein. We need computational analysis to interpret the spectra. The MSMS spectrum obtained experimentally is compared with the theoretical spectra generated from known protein sequences stored in databases such as UniProt and SwissProt. If the experimental fragmentation pattern matches a theoretical peptide sequence, we can identify that peptide. Once multiple peptides are identified, they can be traced back to their parent proteins. This allows us to identify proteins even when the original sample contains a very complex mixture. But identification also needs to be statistically reliable. That's why proteomics workflows use confidence measures such as the false discovery rate or FDR. For example, an FDR threshold of less than 1% is commonly used to control the proportion of potentially incorrect identifications. So this computational stage transforms raw mass spectrometry data into meaningful biological information. So let's put everything together. A complete LC-MS MS proteomics experiment can be visualized as a chain of connected steps. We begin with our sample. First comes sample preparation. Then the proteins undergo trypsin digestion, producing peptides. The peptide mixture enters liquid chromatography, where the peptides are separated over time. The separated peptides then enter the mass spectrometer for MSMS analysis. The resulting spectra are processed computationally and compared against protein databases during the database search. Finally, the identified peptides are used to determine the proteins present in the original sample. So the complete journey is sample, digestion, LC, MSMS, database search, and then finally protein identification. And that is the fundamental workflow that allows modern proteomics to turn a complex biological sample into detailed information about its protein composition. In the next part of a deeper proteomics course, we could go further into quantitative proteomics, post-translational modifications, protein-protein interactions, bioinformatics, and applications in medicine, biotechnology, and food science. Thanks for watching, and I hope this gave you a clear picture of how proteomics works.